the Smithsonian Institute in UC. Nick got his PhD at UC Berkeley and did a postdoc at UBC in Vancouver. Important to highlight that he's Canadian. And he's been in his current position as the curator of marine mammal fossils at the Smithsonian's National Museum of Natural History. Since 2008.
Nine.
And he has a really, his research and publications are really extensive and diverse. They focus broadly on different adaptations, morphological, physiological, and sensory adaptations of both extant and early marine mammals, and sort of link these with all kinds of different aspects of life underwater. So behavioral, predator-prey interactions, life history, evolutionary diversity, so all kinds of different things that he's looking at. I think I'll leave it at that for today, pass it over to Nick, but I think you're really going to enjoy his seminar here today.
Thanks, Lesley. Is there a chance to dim the lights in the front?
I'm going to do that. There are also some seats up front if people would like in the back to see the pictures better.
Okay. I'm really happy to be here today. Thanks to Lesley for the introduction. I wanted to take this opportunity at the seminar to kind of have it complement some of what I shared yesterday in the class lecture for marine mammals, where it was just a broad overview with when we were talking about the evolution of marine mammals, which I think is one of the more interesting aspects of any question that has to do with the origin of these top consumers in ocean ecosystems. So I've kind of, this title for the talk is not disingenuous, but it's intentionally very broad so that it captures a lot of the stories that I want to share. And one of the major stories is this story of the discovery and understanding of the world's largest fossil whale graveyard here in the Atacama of Chile. So that's what that picture is about right there. But I do want to step back and just kind of frame why we should care about fossils in the first place. Why at all does a fossil record matter? If you looked at the modern world, and if we want to understand where whales fit into the family tree of mammals, one easy way is just to look at their DNA. And if we just sampled all the extant species, you would say, well, whales nest in with other, they fall in very close, they're sister taxa to other even-toed, hoofed mammals. And so those are all going to be mammals that we know and love and eat. Cows, pigs, camels, deer. Hippos, it turns out, are the most closely related to whales. And so when you line all those species together, the one that's not like the other is definitely whales. There's so many differences. And that profound amount of morphological difference, let alone ecological, tells you something about evolutionary history. There's a lot of evolution going on with whales. And this is where the fossil record tells you information that you wouldn't otherwise know. And that's really the big lesson that we get from any view about the fossil record, is this is information about deep origins that we wouldn't otherwise know if we just looked at diversity in the modern world. And that has to do with the fact of extinction, but it also has to do with evolutionary transformations and changing ecology through time. So those are two big messages that I really want to hit across. Besides the fact that what we know about the early history of whales, all these names in gold here, that helps resolve these early transformations that tell us how whales became the giants and the really important ecological consumers in the oceans over the span of about 50 million years. That early evolutionary history, the transition from land to sea, going from terrestrial ancestry, and the challenges of adapting to life in the water. There's both constraints and incredible ecological opportunities. That story is written in the fossil record, and what we can tell from these changes, looking at these skeletons, going from animals that looked like they really had no problem living on land to subsequent transformations to the skeleton that allowed for an increased ability to survive and thrive in ocean ecosystems. So a lot of that story has to do with what we see in the first 8 to 10 million years of whale history, this land-to-sea transition. And it happened during a time period we call the Eocene. I'm not going to go heavy on geologic time periods because that kind of bores people, although it's part of my life. I like to think of this slide as Eocene beach party right here with all these early whales, all these different evolutionary experiments, branches on the family tree that are now extinct. They're all pursuing different kinds of ecological ecomorphs that some of which we would recognize today in this artistic reconstruction, which is a little bit apocryphal because they didn't all live in the same place at the same time. It's more kind of an aggregate here. But we certainly know from the fossils that we do find, early whales were experimenting in these different ecological modes. Some look like sea lions, some look like crocs, some look like otters. So they're all doing different things. They all went extinct, and the only ones that begat anything are the ones that became obligately aquatic. They divorced themselves completely from the land in terms of life history. And then we can look to the modern world. Some 40 million years later, they now, in many cases, have become ecological consumers whose influence is disproportionate to their abundance. One whale, many fish. We think that some of them are even able to mediate top-down ecological trophic cascades. That's a really provocative idea that I think has upheld since the time that it was first articulated about 15 years ago. So the question I have is, how do we go from this to there? That's a lot of evolutionary change in about 40 million years. To answer that, you really need to know what's going on in this time period since the Eocene. And to do that, you have to go out and find the fossils and actually understand what they mean. So we do want to really look at, in no small part, their changing ecological role through geologic time. So this is where there's a lot of blend of ecology and evolution in paleobiology. I'm really fortunate to work at a place like this. You can walk off the National Mall in Washington, D.C. and go into the Sant Ocean Hall. And there you can see this great, beautiful exhibit about whale evolutionary history that takes you back in time from, we have a beautiful North Atlantic right whale reconstruction, all the way back to the earliest whales here, Myocetus, which had 4 weight-bearing hind limbs, and the transformations that go to this beautiful, much younger skeleton. This is Basilosaurus. It sounds like a dinosaur because when they first found it, they thought it was a lizard. But it took Sir Richard Owen in London to realize that that was not a reptile of any kind. It was actually a mammal. And of all mammals, it was more closely related to a whale. So in 1844, Richard Owen recognized that this was actually not at all a reptile. We'll still call it Basilosaurus because of the rules of nomenclature, but it is an early whale. And so since that time, we've really filled in a lot about the early evolutionary history of whales and have a much better understanding. And it's a story of geopolitics, too, because there's some places we can't go back to anymore where we found a lot of their early fossils. Myocetus is from the Baluchistan hills of Pakistan. So we will not get any more of Myocetus for some time because of theaters of war and other geopolitical reasons. But yet, if you do the work, if you're able to be lucky and really grind through it, fossils can tell you a lot about evolution and evolutionary history that we wouldn't otherwise know. And at the Smithsonian, we do have many of these fossils. So I really have my work cut out for me. Fortunately, we have a great modern collection, too. So there's an excellent comparative basis for addressing these questions. But there's always the need to go out and if you really want to know about that story of how whales became rulers of the sea, in no small part, you do need to go back out into the field. And so when I go out in the field, this is really sort of my philosophy out there. You're wearing 2 hats. You're wearing a geologist's hat and a biologist's hat. But it's really important to think about walking through these ancient environments as if you are a biologist, as if you're doing a transect survey. How can you know? And there's challenges because doing geology means that you have an imperfect record before you. There's going to be biases. There's going to be glimpses that you cannot know about, even if you have the direct evidence. So it is very much kind of a Sherlock Holmes situation where you're doing detective work. Something happened, we do have evidence, but we don't know, we don't have the entire record. So you do have to make inference. You have to use all the tools that good detectives use. And I think that's important. What this picture is, this is at the end of the day, this is just a random piece of whale rib, but those scratch marks on it, those are shark teeth bites. That whale rib was either scavenged or actually eaten in life by a shark. Hard to tell the difference. Maybe one grad student will eventually figure out how we can know. Was it worth collecting? I think I did eventually collect it. Not that scientifically important, but it does tell us about ecological interactions through time. And that's what's really cool and important about that. So doing fieldwork, how do we actually go find these fossils in the first place? Plate tectonics is driving a lot of what we understand, a lot of field programs, because in some cases they are literally uplifting ancient seafloors into places where they are now accessible. Those ancient seafloors here off the coast of Chile, we have the Peru, Chile, or the Atacama Trench, the subduction of the Nazca Plate underneath the South American Plate. that happens imperceptibly, in an imperceptible way, but occasionally we actually can recognize it when it sets Santiago rocking. There are big earthquakes along the coast of Chile. That happens because of this slip strike between the two plates. Over time, it will raise what was once seafloor into the Atacama Desert. That's a great place to look for fossils because it's arid, dry, all those layers of rock are just open. It's tremendous. And so this is a great opportunity to actually address these questions because we have a lot more access to the record of life in the Humboldt Current system over millions and millions of years right there in Chile. And this is the other reason it's really interesting. The past telling you things you wouldn't otherwise know. There are completely extinct groups of marine mammals we have no record of today. One of them are marine sloths. Off the coast of western South America, there's not just one or two species. There are 5 different species of aquatic sloths, a whole radiation. And what's really neat, they, for the most part, look like a big furry sloth that you might see in Central America. But over the course of those 5 species, the youngest ones show the most extreme hallmark adaptations of adaptations to marine life. So we see thickening of limb bones and ribs. We see elongation of the muzzle, changes in the dentition. And when we look at the isotopes, it's telling us a very clear story. These are feeding on seagrasses. There are no seagrass habitats anywhere along the coast of western South America today, except for one small harbor that still retains seagrasses. And I made a special trip out there, and it was just a small port town, but they still have seagrasses. Porto Aldea, I think, is the place. So that's a relictual habitat of what was probably once much more widespread because we find marine sloths all the way up and down the coast from Peru all the way to Chile. Another really weird thing we do not have today, walrus whales. There are no walruses, which are benthic suction feeders in the northern hemisphere. No walruses at all today in the southern hemisphere. That niche is unoccupied. 5 to 3 million years ago off the coast of South America, there is a dolphin that evolved a walrus-like face and even has tusks too, asymmetrical tusks. But it certainly maybe even wasn't an echolocator, but by all accounts, when you look at that morphology of the face, that shortened muzzle, downturned snout, a vaulted palate, those are, again, traits that we see with walruses. Odobenecetops, the walrus-faced whale, its name even reflects that ecological convergence. So there's a lot going on in South America that's very strange when we look at their fossil record that we don't see today. That makes it a great place to be asking these kinds of questions. So this is what it looks like when you are wearing both hats. That we're definitely wearing our geologist hat, much more so than our biologist hat here. This is the Atacama Desert as it goes out to the coast. The Humboldt Current is right off to the right there. But these sea stacks are abundant. In coastal deserts we find at these latitudes all around the world, whether you're in Australia, whether you're off the coast of Namibia, Angola, South Africa, Chile, Peru, and in the mirror to California in the northern hemisphere, westerly coastlines have the right kind of setup that not only lead to upwelling systems, but also the exposures that we need to actually understand how those systems have changed through time as we can read through the fossil record. So here we're going through we're going through different rock sequences that are telling us about changing environments through time. They're not all the same rocks everywhere, but if you're lucky, you will find fossils there. You need that context, and so that's what we're just doing, walking up and down these cliffs, trying to understand the layers of the rock and how they relate to one another. It's challenging because of plate tectonics. There's abundant fault lines all throughout the place, so you're never getting the same record as you go north to south or east to west. You need to be able to assemble that book with good sedimentologists, good geologists. So a lot of disciplines are involved in just the simple act of going out into the field and trying to reconstruct an environmental history in which you can actually place that biological context. And so this is, I love this slide because this is the moment of discovery. This is when you get really lucky and you find a fossil. This is now Dr. Carolina Goodstein. She was a grad student at the time. Literally 30 seconds after she found the ear bone of the fossil whale, those two pieces get put back together. And there was a whole skeleton there. She spent the rest of the afternoon digging it up with her students. But this is what's great. This is about the arid desert environment. Fantastic exposures of rock, lots of erosion going on. Not many people, no blacktops. You don't have to worry about cars out there. It's just miles and miles of terrain that's completely available for prospecting. It's slow. It's tedious at times. It's random, and you have to be lucky. But sometimes you can be really fortunate. And so this is where I just want to tell a side story about a very lucky discovery. And it starts off with the man at the far right of this picture. His name is Mario Suarez, but he goes by the field handle Tuareg. He thinks he's kind of a desert rat nomad, named after the Moroccan tribes. He only wears orange, so he's only wearing an orange jacket there, but most times he has like an orange T-shirt. He's a very quirky fellow and prone to embellishment. When I show this slide, I usually try to highlight what's great about doing fieldwork is you get to coordinate not just with colleagues from different countries and different cultures. This cuts across national institutions, national museums, universities both in the United States and in Chile, grad students, professors, curators. It's a great bunch of people. So Mario, on the far right, prone to embellishment, kept on telling me about this one site a little farther outside of our actual field region where we're doing a lot of this field work. He said, there's so many whales there, you really have to check it out. And I had remembered going there before there was some new construction going on, and I'd seen a few of the fossils that he was talking about. These are mainly, this is a road cut of the Pan American Highway, and I'd seen a few whale bones in the side of the road, and I said, okay, well, there's maybe one whale skeleton there. It's going to be hard to excavate, probably not worth our time, because it's far away from our main field area. But he kept on telling me about all these whales. And it's my fault for not having believed him initially, because he is prone to exaggeration. When we went back, this is in the fall of 2011, he said, you really have to see it. So we went out, and he rolled back every tarp, because the road construction company that was tasked with expanding the highway, again, geopolitics is playing a role. Chile is becoming an active economic power in that it has mineral resources that growing economies like India and China absolutely need. Iron ores. Maybe some of you have seen the movie 33, about the 33 miners buried in the copper mines near Copiapó. Copiapó is 1 hour down the road from this fossil site, right on the Pan American Highway. This side of the highway was being expanded by the road construction company, which was adhering to Chilean law by running an environmental impact statement. But they then decided they had complete rights to go through and expand. They kept on finding one whale skeleton after another. And every other black tarp going into the distance, this is just on the north side of the quarry, is another complete whale skeleton. This one is complete nose to tip. You can see the skull right here, jaw bones, rest of the skeleton going down to the tail, even the flippers. Every other black tarp is like that. And this is just the north end of the quarry. The south end of the quarry has many more skeletons. So I saw this, and I didn't know what to do, because I don't wish a whale skeleton on anyone. It is a huge burden. Many, many museums have tried just to salvage a whale. You know, whales wash up along Long Island. That's a big problem. Nobody wants to deal with that. And the same is true for a fossil skeleton, because it takes a lot of work to excavate them, and then you want to do it right, and you want to know what it is, and you need to be able to store it. There were over 40 skeletons of fossil whales, all perfectly complete, all this size. So I didn't know what to do. And we were at the end of our what turned out to be our first field season, and I didn't have an answer. But this is where circumstances, for me at least, became fortuitous. At the Smithsonian, there was a nascent program in 3D digitization. So it was the ability to capture a three-dimensional model, digital model of an object, was really getting off the ground, and they were looking for partners at other museums. And I had remembered meeting them randomly in the hallways of my department because they were doing a demo, and I didn't think much of it at the time, but when I saw this, I made the connection in my head, and I said, you know what, I wonder if they would be willing to come out to the Atacama, if I can get enough funding to come back and digitize this. There's no guidebook for doing this. This was really improv. But I thought that they could help. So I got back to DC, and we were all overwhelmed by the scale of the problem before us. Because there's a key scientific question driving this, right? Why? What's going on here? Why do we find this density of complete whale skeletons? I've never seen anything like that. Anything that rivals that is not this complete, not this dense. What is going on? Within 2 weeks, we did the best we could, and we were back. This is the same place 2 weeks later. You can see, keep your eye on this skeleton. It's almost standing in the same place. So we erected tents around the best skeletons, and I'm in the back doing something really important on email back there. But the two guys in the foreground, they're what I end up calling the laser cowboys. They worked so hard. They're program officers of this new 3D digitization department. I was just talking to somebody before the talk. They're now scanning one of the space shuttles. They scan the Apollo 11 command module. They're going throughout the Smithsonian. We have over 136 million specimens collectively at the institution across all of our museums. You can't really digitize every single one of those. That's going to take 1,200 years to do. But instead, we really want to digitize 3D in a very high-resolution way. The specimens that we hold in trust for the nation, everybody here, that tell stories, that tell compelling narratives. And so fortunately, they weren't busy enough at the time that I could bring them out into the field. And we didn't have that much time because the road construction company had to complete the task of removing every single skeleton. And they were jacketing these entire skeletons in whole, in plaster jackets that I've never seen that big. Even for dinosaur skeletons, you wouldn't do them that big. It was insane. But these companies have a lot of resources, and they were hiring Mario's volunteers at the local museum to help doing this. So this is why we did this. This is why we used long-range laser scanners, laser arm scanners, because we wanted to capture that precise geometry, the fine resolution detail of each skeleton, as many skeletons as we could, to be able to answer that scientific question. The arrangement of the bones, their connection, the texture, the orientation, the all sorts of details that we may not even be able to see with our eye, but may be captured by something as precise as this micron-scale laser arm scanner. That buys us time back in the lab to actually process these and hopefully try to answer that question of why. The whole study of how things die and fall apart, taphonomy, is what we want to, that's the discipline that we're applying here. We wanted to apply that with the digital data because we had no time with the real data. So we worked really hard. This is what the southern side of the quarry looks like. By the time we came back 2 weeks later, many of the skeletons had been removed. Each black tarp was saved for us. You can see these 3 skeletons here, actually. That's going to be a reoccurring motif. Keep that in mind, because I'll talk more about that in the skeleton. That's especially compelling when you have 3 skeletons lying on top of each other in a logjam. Keep that in mind. But the scale of the site was really spectacular and extraordinary. So our Chilean colleagues, you can see the South Pacific Ocean out here. This has been uplifted about 60 meters over the last 7 to 9 million years. That's what good geology told us about the site. Our collaborators didn't know if we were going to get back in time. So they did start to deploy what has been a standard technique in paleontology for over 150 years, which is make a latex peel. And then you have latex under here, a plaster mother mold jacket, pop off the mother mold jacket, peel off the latex, and you have some kind of replica, a mold, that you could then fill with plaster or resin and replicate what's under there. The problem is that can be a very destructive process, especially if the underlying bone is porous. It will catch on the latex. You stand a chance of jeopardizing the original specimen. So it's not ideal. It's actually better used really only in trackways. But that's, again, not exactly the same situation here. They didn't know if we were going to be able to come back in time. So they did start to do this on one of the whales. These are just more field pictures because I think it's fun. I think this is part of the logistics that people may not realize about doing paleo field work, and especially in this way. So the laser cowboys started working immediately as soon as we got off the plane. The added challenge, so this is why they were building another lane of highway. This is midday. This is why they're expanding the Pan American Highway in this empty part of the Atacama Desert. There's just simply too much economic activity going on to hold one 2-lane road. So, but when you're doing funny things by the side of the road and a bunch of gringos working with Chilean colleagues from the capital, that will attract attention. So before we knew it, we had media all over us. And if you are trying, I don't know if anybody's ever done this, tried to do science with somebody with a camera right over your shoulder, it's not so much fun. And so very quickly I realized I had to also control the media side of this. They wanted to know why too. That's a very clear question. What's going on here? Why do you have all these fossils? What are you doing inside your black tents? Why do you need lasers? So it's important to handle that because there's the potential that you could ruin your own story. You know, if you think you have a great answer to that, you don't want to give them the answer because that jeopardizes the embargo process that can happen with scientific journals. So this is what a good grad student is useful for, is to go take them on a tour, get them away. And so that's just an added challenge that this kind of picture reminds me of. And we also didn't get along so well. I mean, we got along great. You can see the 2 sleeping bags in the background here. These guys were sleeping on site because we had so little time. And they were working really, really hard. This is just a lively discussion about how to move forwards with the resources we had, with time available. There was no guidebook for doing this. It was definitely fueled by chocolate and coffee down here. But this is, again, a fun picture which reminds me of science happens in a social context, and you have to have trust with partners who may have very different backgrounds. Both of the laser cowboys are really exhibit artists by training. They're not really scientists. So this was a very special, multidisciplinary problem that we addressed. Scanning day and night, this is a time-lapse picture at night. The nearby town of Caldera and its port are lit up. Again, these 3 skeletons that you can see. I think I have another one of these. Yeah, and there's Vince, the self-portrait. This is probably at like 2 a.m. And here's what's interesting. This laser scanner, long-range laser scanner, we brought it from North America. It was one of the key pieces of 3D scanning that we used. It had to be hand-carried on the plane. Other things were shipped. It turned out that through no fault of our own, it had a Hubble Space Telescope problem. One of the mirrors was off. So in collecting 360-degree data, we couldn't get complete alignment of the 2 sides. And so we needed a contingency plan. And so a lot of the 3D data sets you'll see are not actually laser scans. They are photogrammetry data sets. Now, 3D photogrammetry should not be too new to many people here. 5 years ago, it definitely was relatively new. And it basically has to do with compiling a nice data set of 2D images that a computer program can then use an algorithm, if enough of the images overlap, to pull out a 3D model. We use that on many of these skeletons. I'll tell people when we use them over here. But it's really important to have contingency plans when you're doing something that nobody else has done before. So this is towards the end of that, 7 to 8 days. It took a long time to scan this. We had our own laptop up here, so you could actually visualize the data. Really important if you're doing something that you only have one shot at doing, back up your data. You want to make sure it looks good. Vince is coming down to the tail of that one complete skeleton. This is really, again, really late at night. Carolina turns out to be a crack electrician, so she wired up all the tents with light bulbs that we got from the local city. So there's a lot of fun side stories to that these pictures remind me of. But this is what that data set looks like. If this was in full size, it would be about from where I'm standing to that near wall over there. It's about 24 or 26 feet long. This is about 16 million polygons. It's a gigantic data set because we collected micron scale data. And so we had, this was a challenge back in 2012. How do we deal with this? It's a very precise data set. It is just a surface topography. We don't get the bones on the other side because they hadn't been fully exhumed. Once they do get prepared and all the rock is removed from bone, yeah, we could scan them again and then drop them into this grid for which the bones were originally put, in their original position. You can see a paintbrush over here for size. This is great. This is what we need to actually address a lot of these key questions about the taphonomy of this site. But we had to develop the digital workflows to deal with it because that was such a large data set and it was so novel at the time about how to use it. One thing we did do, you can colorize it. That makes the bones pop out a lot more. And we were fortunate in having corporate partners who got really excited by the scale of the data set we had. They helped us print it in 3D print. This went on the wall after we published our findings on the story. So that happened about 2 months later. These are a lot of the original field teams here at the Smithsonian. You can come to Washington, D.C. and see this for yourself. Get really close to a full-size 3D print of one of these whale skeletons. It was something that I was really skeptical about to begin with was 3D digitization. I thought it was just shiny Hollywood magic and not really that important until I realized that it really can do 2 things. One is answer scientific questions, and 2 is braid the 3 things that we do in museums, which is research, collections, and communication. So that's a research object in that there is real data there that we wanted to measure and know. It's a collections object because we printed it. We can always print more because we have a digital copy. The real doesn't make a substitute for the real thing, which is stored in a museum in Chile right now. But when it has a physical manifestation, then it becomes a collections object. It has a catalog number at the Smithsonian. And then the third subpart, communications, it tells a story. And it can tell a lot of different stories. It can tell stories to different kinds of audiences, no matter what the background is. So I've really become much more of, not a believer, but I understand why 3D printing can become important in informal education settings, especially like museums. So, it's not worth talking about, but so this is what's fun is you can print a whale and hold it in your hands. I'll pass out this one. This is actually the same print. Here, I'll start down here. Just make sure it gets back to me. I think I have another one too to pass out. Oh yeah, this is a smaller one. Here, Lesley. Pass that out. So it's fun to be able to, this is, the scale bars on this 3D print are 20 to 30 centimeters. So that gives you a sense of the scale of that individual skeleton. It's pretty big. These are the jaw bones here. This may not, at the time that this happened, I thought it was pretty eye-opening, and it still doesn't get old for me, actually, is being able to hold skeletons like that. It's a facsimile. It's not the real data, But we are still visual, tactile creatures. That is one way that we understand the world around us. And it helps because the scale of a whale skeleton is absolutely so large. Having it in the palm of your hands, that really changes your ability to understand it, changes your ability to ask questions about it. And I'll get to the questions in a second. The other really neat thing is that we really worked hard with these digital workflows to be able to create a platform to share them at. So that platform is now Smithsonian X 3D, 3d.si.edu. If you have, it's okay to open your laptops if you want to go there right now. This is a QR code. You can probably type this out really fast. I'll leave it up here for a while. You can then go see that model. If you type in that web address, you should go to exactly this view. And that's the power of this 3D viewer, is that it will visualize the 3D data. You can measure it. You can change the lighting settings. You can then log in and download a 3D model and print it yourself. So this is a way to communicate at a scale that I think is really, really valuable and really important, because it lends transparency to the process of what we're trying to do. Those whale skeletons that we scanned, anybody can download and print them. Anybody can measure them. Anybody can then actually check our scientific findings. And I thought that was really important. The day we published our findings, which I'll share with you in a second here, any 10-year-old in Kansas or in Chile would be able to access this, especially in the town of Caldera. It's their national patrimony. They keep the fossils. We're able to render and visualize the 3D data. I think that's really important for communicating the value of the real thing, and also for spreading the visibility of why we ought to care about the deep past. Even if you can't see the real thing, you can see a very, very good facsimile. You can print it too. It's really changed how I think about things and how I do the science that I do. So, okay, what do we know about what's going on here, and what can we say about all these fossil whales at the site? So this is a photogrammetry model, 3D model. It's almost as if you were up in a crane or a hot air balloon over those three skeletons. You can see one skeleton here, nose to tail, and then a much smaller one. This is a juvenile. So the Chileans took to calling this La Familia. I'm agnostic about whether or not it's a real family. I don't care, because these data are tremendously compelling from the standpoint of being able to test the questions we want to test about how the site came to be. The skeletons are incredibly complete. There's not much scatter from any individual skeleton along the midline axis to the bones that are farthest away. You can see finger bones over there. There's a hip bone underneath that vertebra. All the jaw bones are there. The skulls are mostly intact. The jaws are mostly in line with the skulls. They're not spread out. They haven't been moved apart very far. That tells us a lot about the physical environment under which these skeletons are preserved. And the details of this kind of model are just spectacular. You can zoom in to centimeters. It's actually a millimeter resolution model from the photogrammetry data. This is great, because I'd like to think that these are all digital data sets that have a legacy. They have durability. They have longevity, so that they can provide dissertations worth of data for future students to do. There's going to be a lot more questions I haven't yet asked about these data. I hope students will go back and do them. I hope just anybody does it. So that's kind of the view about the digital data. Okay, what else is interesting about Cerro Ballena? Something I didn't tell you, but you might have been able to see in this picture, is that there's not just one site. It is not one rock layer. It's actually 4. And you can see that with this tarp up here and that tarp down there. There's 4 different levels. This view is just the top 3. Those are 4 different bone-bearing horizons. They all have fossil whales in them, and they're all complete. And there's approximately, in this stack of rock by the road cut, which you can't actually see in any one place. It's a composite picture. You have to walk up and down, and it took us a while to figure this out. There are 4 different levels. So that tells us there's 4 different times. Minimally, by our estimates, between 10 to 16,000 years of history is recorded there. It's essentially the same environment. I'll tell you more about the environment in a second, but you might guess that it's not exactly a very active place. It's probably pretty quiet. I'll tell you, it's also not the place where whales tend to live, but tends to be where they die. The other finding is that it's not just large baleen whales there. It's dominated by large baleen whales. We found over 10 different large marine consumers. We found no invertebrates. We found the traces of invertebrates. We found trace fossils. But we found not just large baleen whales. We found 2 different species of seals. We found Otobena sea tops at this site, the walrus whale that I'd mentioned before, that had only previously been found in Peru. Lo and behold, at this roadside site, we found the skeleton of Otobena sea tops, 1,000 miles away from where it had been known previously. We still haven't published on it, but we did mention it in this paper. And there's also an aquatic sloth from this site, amazingly enough. So in 250 meters of road cut, across 4 layers of bone bed, we're picking up everything that we have found in Peru out of the result of 30 years of work in that country, right here in this road cut in Chile. There's also billfish. These are all high-level trophic consumers, whether it's a pelagic fish or marine mammal. And what's really interesting with the large baleen whales, we're actually able to look at a lot of the data about not just their scatter, not just their length. We can also talk about how articulated they are. We can also say something about whether they are preserved belly up or belly down. So whales preserve belly up, while not exclusively, but you might argue that that would tend to indicate the whale was dead prior to burial. It might have also been nose up, blowhole up, and dead, but with very large rorqual whales, which have this big ventral throat pouch that serves almost as like a life preserver flotation device, when they die, they do end up belly up. You can see a lot of pictures online if you just Google dead whale. They tend to be the large rorqual whales. They're belly up. And so that is actually telling you something very important about how they died and how they got buried. This is that detective. There's something happened here. We want to know whether it was a murder, whether it was a graveyard, what's going on. So these are all the kinds of data, and this is going to help us test those different ideas. Okay, something about the depositional setting. What was Cerro Ballena? I said it wasn't exactly the place where whales lived. It turns out to be a tidal flat that's occasionally emergent. That's a very quiescent environment. It's fed with a little bit of fresh water from a nearby river. This is kind of what that might have looked like back in the Miocene. This is 2,000 kilometers south of Cerro Ballena near Chiloé in Patagonia. This is our sedimentologist took this picture and sent it to me in the email. He said, "This is what Cerro Ballena looked like, just so you know." And you can kind of see that in this paleogeographic reconstruction we have here. Cerro Ballena being at the north end of what was once a very large embayment, sheltered by large promontories from what were probably rocky seashores. But the overwhelming current direction coming off the Proto-Humboldt current was pushing waves in that direction. So we do have kind of a setting that was quiet, persistent, and it was a good catchment area. Another key piece of evidence is that, like I said, the large whales are belly up. And I started to home in on harmful algal blooms as a good explanation because, in science we like the simple explanations that have broad explanatory power. I started to think pretty quickly that what we have are mass strandings. And we have repeated mass strandings because we're finding them across 4 bone bed layers. We find the same data set across 4 bone bed layers. Harmful algal blooms, many different taxa can produce poisons that get amplified up trophic levels in marine communities. These are diatoms, these are dinoflagellates, cyanobacteria. They will all produce toxins in some quantity or another that eventually become so biomagnified by the time that they're ingested by a mammal or a consumer at that body size, they can become neurotoxins. And so there's this great example, not too far from here, off of Cape Cod, 1987, 1988, some 24 humpback whales were killed more or less suddenly by ingesting mackerel that were completely loaded with red tide, dinoflagellates, that didn't necessarily kill the fish themselves, but at the concentrations that the whales were eating them, it was a neurotoxin and it causes asphyxiation. So these whales died, in some cases, within 90 minutes. This one whale was seen on a whale watch, and 90 minutes later was on the beach, belly up. So that to me is pretty compelling data, especially when we look at not just the density, so it includes both adults and juveniles are included in this, which is consistent with that La Familia picture. You get different body sizes, many different species, it turns out. If you read the report about this, they talk about not just humpback whales. There's also sei whales that were involved, and maybe a Broudy's whale, multiple species. So any explanation, so that's what makes harmful algal blooms compelling to me, is that it explains multiple species, it explains the condition of the skeletons as they get to the site, and it is also a known phenomenon that's happening off the coast of Chile, even today. So if this is true, is there a smoking gun? And we always love smoking guns in paleontology. You probably heard about that for the mass extinction at the end of the Cretaceous that killed the non-avian dinosaurs. We collected tons of sediment, spent a year under an SEM microscope. The best we could get were that we'd find these little spherical structures about 20 to 40 microns across, covered in iron. This is about the right size to be a dinoflagellate cyst, but there's no morphology available because the environment has been so changed over the course of fossilization. We're just not picking up that morphology. But they are loaded with iron. There's iron throughout the site. And iron is a limiting agent for harmful algal blooms red tides today. And some of the most iron-rich mountains on the planet are the Andes. Ferruginous runoff from the Andes, we think, is a pretty good explanation for empowering harmful algal blooms off the coast of Chile. It's a phenomenon that happens today, and it's likely that it happened in the past. So we pitched, this is our idea, that we have repeated mass strandings of not just whales, but marine mammals. This is the density, color-coded by stratigraphic level along the site. And we think it's a pretty good explanation, because the other explanations that can be advanced, and if you guys have explanations, because I know there's oceanographers out here, send them over. But what we want out of any one explanation is broad explanatory power for the range of species we see, the repeated nature. So that catchment area at Cerro Ballena, that was a nice tidal flat for catching whales and other pelagic consumers that died at sea. We're also getting a nice coastal signal, because we're getting pinnipeds, we're getting seals. Those are probably pretty close to inshore. They probably died relatively suddenly, which explains their nice condition. And the fact that it's happened many times, we find mass strandings in the same place over many years. So it stands to reason that we should see this in the fossil record. This is probably one of the best examples of that. We can talk more about the science in a second, but when we publish, so not only do we have the 3D models online, we created a website to house the 22,000 digital images that we collected through the course of fieldwork at Cerro Ballena, not just in 2011, but all the way to 2013, 2015. That was really important because we wanted to make open access automatic for the kinds of data that we were presenting for this very reason. I wanted other people to be able to test this. And so all those were available the day we published it. It was a lot of hard work to get to that point. But I'm really proud of that because that is should be one of the outcomes of international collaborative work. We want to make sure that everybody makes sure they feel like they are getting respect, getting access to the data, and able to enjoy the visibility of the result. So what's happened with the physical? I've told you about the digital data. You're seeing the manifestation go back. What about the fossils themselves? This is more or less what it looks like today. This is an old Victorian train station in town at Caldera. This is where the Natural History Museum is, for what it's worth. And you have jacuzzi, piano-sized plaster jackets that house all these fossil whales. That is the physical legacy that needs to be dealt with. Preparation, mechanical preparation. It's going to take decades, even if you have enough technicians. This is a problem in North America. It's not just a Latin America thing. It is a problem here as well. Not enough people working on the fossils. But fortunately, I don't think that's prevented us from answering the big questions about Cerro Ballena. But it does mean there's probably a lot more fun stories to find out about from those fossils alone. So I don't have too much. I have about 15 minutes. I'm going to try to explain a few more things. That was the Cerro Ballena story. The result of that is that changed my view about how I should do paleo fieldwork, in that documentation can now be done with 3D data. That's really changed how I think I can address different field challenges, projects, and problems. This was a site in Panama along the Caribbean side where I got a phone call one day from a colleague at the Smithsonian Tropical Research Institute who said, I think we found a fossil dolphin. Here's a few pictures. Do you want to come get it next week? And I don't teach classes, so that's actually something I can do. Hop down to Panama. And so we were there 7 days later with my grad student at the time, but now curator of marine mammals at Los Angeles County Museum, Jorge Velazquarbe. We're pulling out the skull that we've jacketed. And the trick was we only had that day because of tidal cycles in this environment. There's only a few times a year that you can actually go out and get that skeleton. And so we were racing the tides, very much so here. We had about an hour left to pull out that skull. I had not yet gone to Cerro Ballena at this time. But what I did realize after the fact is that we could use 3D data to actually really change how we describe these fossils. That's a picture of the actual skull. Turns out it's a relative of the Amazon river dolphin, found in Panama, found in marine rocks. So that's telling us a lot about marine to freshwater transitions, and that's something cool that we see in the evolution of river dolphins many times in toothed whales. But these beautiful images that look like lithographic art, that's digital data, and that's all available online with the description of this taxon, Isthminia panamensis. We also did that with Albacetus oxymictris. This was a project by Alex Boersma, who was a research student. She did this for her honors thesis at Vassar College. This is the description of a fossil whale skull, sperm whale skull, that had been in the Smithsonian's collections for 90 years. Misidentified. A lot of the professionals knew about this skull, but nobody had yet been able to tackle it because the skull weighs about 400 pounds. It's almost impossible to be able to study it in that way. You can see these large holes. These are the lower teeth. Alex is holding one of them. There's also upper teeth, too. That's totally unlike any living sperm whale. And so it was well worth description. It turns out to be a new genus, new species. And so we did do a 3D model of that, too, and that can also be visualized on 3d.si.edu. So it's really changed how the kinds, being able to have this kind of platform for specimens in the National Museum and being able to digitize them really opens up the door to different kinds, being able to tackle different kinds of projects, whether they're just simple species descriptions or they're more paleoecological, like Cerro Ballena. So I just want to finish by getting at the bigger questions of why we should care about whales. And one of them has to do with understanding, most clearly, going from small dog-sized animals to giants of the sea. That's telling you about one major thing, one big thing we can pick off right there, body size changes. Whales are not just kind of big, they're the largest vertebrates ever to evolve in the history of life on Earth. No single sauropod dinosaur was larger. They might have been longer, those sauropod dinosaurs. You can see one on display at the American Museum right now. That titanosaur from Argentina, it's 110 feet long, but it certainly didn't weigh as much as the largest blue whale that we know of. Those blue whales from the southern oceans that were killed during the heyday of southern whaling in the 1920s and 30s, they probably were over 100 years old. We have no idea how long some of those big gulp-feeding whales lived. But at the time when they were first hunted in the southern oceans, I bet you those were decadal-plus large whales. Some of them that were over 100 feet long. The reliable records we have for their weights, 250,000 pounds. There's no dinosaur that weighed nearly that much. And that tells you a bit about the differences between evolution of life on land at those size extremes and life at sea. So how did whales become ocean giants? Well, we can actually plot out their body size, and this is a collaborative project I've been doing recently that hasn't yet been published. This is the range of body size we see for living baleen whales, from the pygmy right whale all the way up to blue whales. That's a mean body size. We wanted to be a little conservative. But what you see pretty clearly is that no fossil whale, for which we know of, even comes close in body size to blue whales that we see today. And this actually falls outside of what we'd model for passive diffusion, if body size is just kind of a random walk through time. It also is not totally explained by any physical forcing that we can directly look at. For example, if it's a temperature-dependent model, and that's a delta 018 curve. It's loosely telling us about temperature in the oceans through time, built out of a lot of great ocean drilling work that has very high resolution, telling us about major changes in the oceans in the last 65 million years. So what is going on with the evolution of ocean giants? From these data, you'd also be able to say, it looks like it's a very recent thing, that body size categories at the size of blue whales, fin whales, even bowheads and right whales haven't ever been accessed in the evolutionary past. That is really different from the evolution of land mammals, where as soon as mammals become major players on the scene after the end of the Cretaceous, no dinosaurs on land, mammals get very large within about 5 to 10 million years. Here it's taken 30 million years for whales to evolve to anything like a blue whale. Why? What's going on? So what I think is going on is major changes in the oceans that are probably tied to the last 5 to 3 million years. And so when we look at the I don't expect you guys to really be able to see this figure. All I want you to see is that there are hot tips of body size, very large body size categories, that are independent. And they all seem to have happened fairly recently. Those clade-wide shifts, which is something we can test with different models, all seem to have happened within the same time frame. And that time frame is the Pleistocene, the initiation of the Pleistocene glaciation of the northern hemisphere. We live at a very strange time in Earth history, we're about to bump out of it probably, where we have two poles that are glaciated. The South Pole has been glaciated for the last 30 million years. But the northern hemisphere, that's only been glaciated since the last 2.5 to 3 million years. The glaciation of the northern hemisphere and having ice sheets going down to Seattle and back up in latitude, not just once or twice, but over 20 times in the last 2.5 million years, that undeniably had major impacts on ocean productivity. And we think probably on the patchiness of dense prey items that would be the targets of baleen whales, not necessarily toothed whales, although there's some cool patterns, convergent patterns there. Large body size confers a lot of advantages, if you think about it. There's predator deterrence. You're not going to be harassed by killer whales when you're at 25 meters in length as an adult. Maybe your calves will, but probably not an adult. There also confers a lot of advantages for long-range migration, and we know that these large baleen whales do move long distances. So we think that this is pretty compelling. This is telling us something about how body size evolved, but it's actually really, quite frankly, raising a lot more questions than answers. You can also look at this ecologically, too, by regions, and this is something I did in an earlier paper this year, splitting it up by North Pacific and North Atlantic, looking at the evolution of really large ecological modes, whether it's filter feeding on the bottom in blue or herbivory on the top. We see that there's turnover in the North Pacific. This is another extinct group of marine mammals called desmostillions. They kind of look like hippos, and they only ever lasted from the early Oligocene to the late Miocene, and only in the North Pacific. We find them from Japan all the way to Baja California and nowhere else. They're probably closely related to elephants. We don't know much about them, but they seem to have occupied the marine herbivore niche in the North Pacific until sea cows showed up on the scene. And that has to do with the separation of North America and South America from an open Central American seaway before the rise of the Isthmus of Panama. You can see in the North Atlantic where sea cows have been since the early Eocene. So that's kind of, this is the center of origin for sea cows and manatees. They started in Jamaica about 55 million years ago. And going back to something I'll mention in a second, they show the same stepwise changes in morphology that early whales show, losing their hind limbs, decoupling of the pelvic girdle from the vertebral column. But interestingly, in the North Atlantic, they never really exceeded, they mostly stayed within the same body size, not accessing the very large body size categories we see in the North Pacific. It's a really different story for filter feeding, but we still think overall that there's something going on in the Pliocene and Pleistocene that's leading to the access of these really large body size categories. So body size is being a useful proxy for understanding something about marine mammals in the past, which we then infer is something about the oceans. Most viscerally, we know the past is different, and this is just another reminder of a paper I wrote with Jorge. Today, we only ever find one sea cow or one manatee wherever we look, but in the past, it was completely different. We find multi-species of sea cow communities all throughout the world. That seems to be the norm. So really, when you're a paleontologist, it's not how weird the past was, but it's how strange the present is. The present configuration for marine mammal herbivore communities is so different from any other time in the last 26 million years. That is what's cool about the fossil record. Okay, one last sort of set of points here. When we think about, this is kind of a classic textbook comparison of an ichthyosaur and a dolphin. You'll sometimes see a shark group there too, and those are supposed to tell you about hydrodynamic form, which is selection for movement through water as a vertebrate. You get a teardrop shaped. This is convergence and evolution. We tend to call ichthyosaurs as dolphin-like reptiles. They're marine reptiles. This is a marine mammal. But ichthyosaurs got started on the scene 200 million years before the first dolphins. So really, dolphins are actually ichthyosaur-shaped mammals. And this convergence is something we see when we expand our view across all vertebrates, all tetrapods. Convergence in marine tetrapod body forms is pretty astonishing. Early whales look a heck of a lot like the mosasaurs. This was in Jurassic World, the hero of Jurassic World, which I didn't like, but and early ichthyosaurs too. This is cool. Decoupling of the pelvic girdle from the vertebral column. That seems to be a common pattern that we see. When we see repeated common patterns, that's telling us something about how evolution works. We see seal-like marine reptiles, and we can just look in the modern world. Recently extinct, this is telling us something about human involvement and shifted baselines. Great auks sure look a lot like penguins. In fact, this genus name for a great auk, Penguinus, sure sounds like a penguin, right? And so this broad view, this is best told with this image. We have the radiation of mammals, and at least 7 times mammals have gone back to the water. Marine sloths, desmostillions, sirenians, pinnipeds, arctoid mammals, mustelids, whales. But if you look over the last 240 million years, there are many, many other lineages. Seabirds, turtles, they all have terrestrial ancestors. And we don't know, for mosasaurs, ichthyosaurs, and all these other marine reptiles from the Triassic, we have a hard time pinning down their ancestry. And it's only because we have such a good fossil record for early whales that we can talk about these evolutionary transformations. We want to know more, and that's why we have to go back to the field to figure this out for early reptiles. This is the mirror image of what, and this is why we have Tiktaalik down here for the first transition. That transformation from sea to land, that's kind of epic, textbook macroevolution. This is another macroevolutionary story, which is the repeated reentry of 4-limbed animals back into the ocean from terrestrial ancestors. Very cool. We see common patterns. That's telling us about how evolution works. One last point. We want to look at success, evolutionary success, as maybe some guide for what's happening in their evolutionary past, but with a view to what will happen in the future in on an Anthropocene Earth in Anthropocene oceans. Whales are clearly much among the more successful, just if we look at sheer living diversity today, just look at the modern world. We can also, I also have another slide with the fossils, but I just want to use this to illustrate the point. We see a really very strong modality here between some clades that seem to be very successful and others that aren't. But when we align this by their ancestry, we get this really cool separation. There seems to be an incipient group of marine tetrapods that haven't had enough time to go back to the water, but haven't really risen in diversity that much. Those are like sea otters, sea minks, anything you can think of that has very low diversity, very recent. Snakes are actually like that too. Snakes have not that really high, that great of diversity, marine snakes, but they have been going back to the water for a long time. So we call that a relic zone. Their active radiation zone, that would be pinnipeds, that would be mammals. And if you started mapping on fossils onto this by using their estimate divergence time, ichthyosaurs, you'll get nice placement of other groups. And how those groups place tell you a lot about asymmetries and evolutionary innovations relative to what we might call success. Evolutionary success, how many species we find. So this is something that, this is another direction that I'm going in with other collaborators about this. This was the last slide I used from the class talk yesterday, which is that knowing more about evolutionary history is not just kind of fun, but I think really relevant for changes that are happening in the world right now. Humans are only going to have a greater impact on and a greater influence on the distribution of living species, these top consumers. We've already removed a lot of them from the oceans, especially with whales. None of them have gone extinct because of whaling. The only whales that have gone extinct seem to be from other kinds of mass habitat changes. The vaquita may be like that too. That's indirect effects from other fisheries. The Yangtze River dolphin, that went extinct because we dammed the Yangtze River. So, but we do know that marine mammals are in that high risk for extinction category. How it's going to turn out in the future, I think, is not as simple as it's portrayed here, because we know that geography is different. Some are generalists, some are specialists. The specialists, especially those in freshwater river environments, that's a high risk category. But something like a gray whale, those may be really big winners in the ongoing sweepstakes of climate change. So this is why I think evolution matters, looking to the future of marine mammals. And that's it.
A couple of questions for Nick.
Yeah, we're kind of. Go ahead.
Thanks very much for a very interesting talk. I was thinking about this harmful algal bloom as, which very interesting idea, but I'm not quite sure why it would kill the marine sloths that are eating seagrass.
So that is great. So what's killing both? So carnivores is pretty clear. They'd be consuming finfish. That would explain whales and pinnipeds. So we know that there's an ongoing harmful algal bloom off the coast of Florida that's killing tursiops, coastal bottlenose dolphins, and manatees. And there's two different pathways for killing them. Bottlenose dolphins are consuming organisms that are exposed to the toxins. Manatees are inhaling. It's inhalation of those toxins that are airborne. And you talk to people who are swimmers, people just swimming in those waters experience the same neurotoxin symptoms. And it's not because they're eating anything, they're just breathing it in. And what else is breathing it in? And it might be that manatees are also kind of on seagrasses consuming a bit of it. That's not clear. But both bottlenose dolphins and manatees are among the dead for these harmful algal blooms. So here's the inference. If aquatic sloths are at all like manatees, which as marine herbivores, I'm going to guess they are, that's probably the likely mechanism.
I have a question, but Mary Silver a number of years ago also related harmful algal blooms to seal sickness on the West Coast. So
yeah, domoic acid, is that? Yeah.
Yeah, domoic acid.
Yeah.
The question is, if you go back to the Oligocene and the Eocene, are there occurrences of mass kills, if that's what these are, of ancestors of whales?
I would love to know. I don't think so, and that's because I think what we've done with this study is show the criteria that have to be satisfied to make that inference. We just don't find fossil sites like that. And it was really happenstance that we found this one. There was a good chance we might never have found it. And I like stay awake at night thinking about all the fossil whales that were destroyed when the first lane of highway was excavated. There are probably many other Laphamillas. There might have been like dozens of walrus whales that were just destroyed when that
so
first.
So there are places like in Pakistan where there are multiple big, rich horizons of whales?
So you can have a rich horizon of bones, but unless you're able to excavate it in this way, you may not know that they're all individual skeletons. In Pakistan, what we tend to find are these environments, fluvial environments, where bones are just kind of like hashes, and the skeletons are not associated. They're not stuck together. You'll find a toe bone here and a leg bone here, and they're not the same size. They probably belong to different individuals that may have been lived very far apart in time. When you see something like La Familia, those are animals that likely lived together. I think it's not so far to say that they probably lived in the same pod if they were the same species. You know, the happenstance of finding them log jammed like that on top of one another, that's telling you real ecologic data. And that's one of the big revolutions, I'd say, in paleobiology is the treatment of the fossil record. Once you're able to address all the biases as recording real ecologic data and not just, well, it's random. There are some cases when you have these hashes of bones, other bone beds. It's a broad spectrum of description where you would say that that's not true. So I don't know. So what I know about the fossil record of whales, Cerro Ballena is the best example so far. I hope we find older stuff.
More questions for Nick?
You mentioned earlier in your talk that the western coasts
Yeah.
are better to find fossils. So how do like the current eastern versus western side?
Yeah. Boy, if I said that they're better for finding fossils, there's a lot of factors that go into finding fossils. Exposure of rock is one. In the Northeast, the Calvert Cliffs of Maryland are some of the best, most productive spots that rival places in California and in Chile. What we have on this coast is a passive margin. And so if you drop down sea level on the west coast, it's a very sharp distinction, more or less. On our coast, it's a long, long slope because we don't have the active tectonism. So you just get this rain of sediments. Over time, it's bouncing back. That's called, broadly, that's what happens when ice sheets come back and lift off of land. So there's a bit of glacial rebound going on with topography on this coast. It's happening in about a few centimeters a year, actually.
But I guess my question was then how did like the eastern and western sides of the continents compare in terms of current diversity and abundance of so like
that is a
if
good,
you had your choice.
If I had my choice of productive places to find, I mean, definitely, the current systems that are productive today probably were productive in the past. And the question we want to know is how long have they been productive? You will not find rocks old enough in South America, except in a few places that are really interesting, as old as you might find in Africa and in parts of the coastal plain of the United States. But those are sampling, but here's the problem is that map of the world, that is how it is right now, but it is not how it was even 3 million years ago or even 10 million years ago. So you get this changing configuration of the continents and plates moving across. Different ocean portals are opened and closed. So if you're playing this game of where I want to look, it's really complicated. And it's complicated by all those factors. None of those factors are like easy to control or just even in an intellectual framework. I do have my favorite hotspots, and they turn out to actually, so the answer to your question is not Northern Hemisphere. We've actually sampled a lot of the Northern Hemisphere. The high latitudes, so the Arctic, would be good to sample more, and I just had a paper 2 weeks ago about an Arctic river dolphin, whose relative is the Ganges river dolphin. So in Alaska, we're finding a fossil relative of something that lives in India today. The whole southern hemisphere is basically a big unknown. We have not really looked in Antarctica. We have not really looked across most of South America. Australia, really, really hard to do field work there because you will die if you go out into the desert. So New Zealand is good, South Africa. So there's hot spots in the southern hemisphere, but it's hard work. It takes a long time to find fossils. Am I answering your question? I feel like.
I mean, your focus is on getting the longest time.
Yep, that's what I want to know. Yeah, not necessarily rich sites. Rich sites are actually a burden. I want grad students to work on those. I want to find them. Okay.
All right, well, I think we should hold any more
Yeah,
questions till
come
lunch.
up.
And just a reminder for the new students, we're going to have pizza down the hall in the Okuba room. So thanks very much.
Thank you, guys.
For an excellent
Oh, can I get those 3D prints back?
I think there's a difference here. That to me, they can't.
Well, if you
So actually, yeah.
It's amazing. There's this zoo right near me, and they had this condor in a cage that was literally about maybe 8 or 10 feet wide. It could not spread its wings. It's the classic zoo that tortured animals. Yeah, well, the zoo is amazing.
I feel better now. One thing after, can I just borrow that for a second?
I thought you were going to stick that on your shirt. Yeah, I know.
Yeah.
Yeah, they were
you get outside all the time, there are maps and diagrams.
You make suggestions. I agree. It's so hard to Okay, good. Malcolm? Dr. Bowman. So this form, just don't say in place I'll send you again. You gotta get the poster down. Yes, 20. I sent you email with the numbers.
So the sound itself is probably pretty common. Yeah, so I bought it.
So we have wave ribbon.
Awesome, please do it.
So I'm like, oh, that's perfect. Loading the size of our That's how it happened. So in work, I've only seen road cut that's 20 meters by 2 meters. The site goes 1.5 kilometers. can see bones still going
So,
down.
oh, right.
Roadside on the other side.
So you can specify.
This is not just one. Yeah, there's many more steps.
Yeah.
So we really have a dinosaur national model.
Sorry, sorry.
No, I'm sorry. We should probably.
Oh, yeah. No,
Thanks
that
so
was
much
nice.
for coming.
Probably.
Yeah. That was good.
Yesterday.
You should have hoarded him yesterday in your class because that was. Oh, yeah.
Like this?
Yes, I
This is
know.
like the subject when I was in Chicago.
Yeah.
This is the kind of
Right.
thing.
I mean, and if you look at the anatomy,
Yeah.
they're very similar in a way.
They are.
It's so cool. It's so
See, you're getting closer to my retirement plans. I'm going to hunt for fossils with the leaf.
Oh, I'm never gonna retire, right?
No, I
I'll
don't.
carry the out like Or a sandal.
Thank you.
Another 30 years, right?
We'll come visit you.
Calculated my retirement.
Yeah, right. Genetically, I'll be a jellyfish in 30 years.
Yeah.